Hello everyone. Today here let's discuss a few questions on buffers and cell cycle. First let's see a few questions on buffers. Buffer, first question, name the buffer systems in the body. This would be a 1 mark question. So, you can just name the buffers, which are the buffer systems in the body, and just one sentence on each. That is, first one, bicarbonate buffer system, which is most important buffering system in plasma, by bicarbonate carbonic acid system. Second one, phosphate buffer system, which is intracellular buffering system. Third one, protein buffer system, which is mainly by the hemoglobin protein, and it is by the histidine imidazole group. Next question, important buffer in intracellular fluid. That is, two are there, phosphate buffer system and protein buffer system. Phosphate buffer system is mainly an intracellular buffer. Its concentration in plasma is very low and pKa value is 6.8. The phosphate buffer system is found to be effective at a wide pH range because it has more than one ionizable group and the pKa value are different for both. Then protein buffer system is by the histidine imidazole group having a pKa value of 6.1. and it is hemoglobin buffer. Third question, that is plasma buffers. So, this is just a repetition of the question in different form. Plasma buffers, you can mention the buffer system functioning in blood plasma, which includes plasma proteins, phosphate, bicarbonates and carbonic acid buffers. So, you can explain all the three, that is protein buffering system, bicarbonate buffering system and phosphate buffering system. Next question, that is the anion gap. Anion gap, you can give the definition and the normal range. That is definition, it can either be, that is the difference between the number of cations versus anions, or it is the unmeasured anions constituting the anion gap. It is due to the presence of protein anions, sulfates, phosphate and organic acid, and normally it is about 12 millimole per liter. Next question, that is coming under cell cycle, that is apoptosis. Apoptosis, it can be a 1 mark or a 2 marks question. So, differentiation and growth is needed for reshaping of organs. This could be done only when the old cells are removed from that area. So, the removal of superfluous, aged and particularly damaged cells is done by the apoptosis process. That is the removal of aged and damaged cells, that is called as apoptosis. Nucleus shrinkage, chromatin condensation, membrane blebbing and step ladder pattern of DNA in electrophoresis are characteristic features of apoptosis. So, you can give the definition that it is the removal of aged and partially damaged cells, and examples, characteristic features are that nucleus shrinkage, chromatin condensation, membrane blebbing and step ladder pattern of DNA in electrophoresis. And examples, apoptosis mediated genes include P53 and retinoblastoma RB. So, that would be enough. Okay. Next question is cell cycle. Cell cycle can be asked up to 4 marks. So, including the diagram, you should explain the different phases of cell cycle. So, first you can give a brief description on each steps, then what is cell cycle, then also include the diagram. The term cell cycle refers to the events occurring during the period between two mitotic divisions, and it is divided into G1 phase, then S, G2 and M phase. The cell division is taking place in the M phase. It is the shortest phase lasting for about 1 hour. The daughter cells then either enter into G0 phase or re-enter the cell cycle when there is necessity for growth and repair. In a normal cell population, most of the cells are in G0 phase, then general metabolic events are taking place in the G0 phase. The interphase is the period between the end of M phase and beginning of the next mitosis. In G1 phase, the protein and the RNA increases. The duration of G1 phase is for about 12 hours. Then in S phase, the DNA is synthesized, but only once. Then the DNA contents doubles and nucleus becomes tetraploid, that is 4 in number. The entire diploid genome is replicated into tetraploid genome. S phase is lasting for about 6 to 8 hours. Next, in the G2 phase, there is cytoplasmic enlargement. DNA repair is also taking place in the G2 space. Proteins, especially histones, are also produced. It lasts for about 4 to 5 hours. The total cell cycle is about 20 to 22 hours duration in, especially in mammalian cells. So, explain each phases and what are the events occurring and how much duration it takes. You can also include this diagram. Okay, thank you.